So on to the main event. It's my distinct honor to introduce our esteemed keynote speaker tonight. And I've had the privilege to cross paths with the remarkable man that will be delivering tonight's lecture several times through the years. And I know him well enough to say that you're in for a real treat tonight. Dr. Francis Collins is a physician geneticist noted for his landmark discoveries of disease genes and his leadership of the International Human Genome Project, which culminated, of course, in April of 2003 with the completion of a finished sequence of the Human DNA Instruction Book. Since 2009, he has served as director of the National Institutes of Health, the largest supporter of biomedical research in the world, spanning the spectrum from basic to clinical research. He's an elected member of the Institute of Medicine and the National Academy of Sciences. He was awarded the Presidential Medal of Freedom in November of 2007 and received the National Medal of Science in 2009. And we are proud to count Dr. Collins as both a member and a fellow of the ASA. Dr. Collins is also the author of a very popular book called The Language of God, A Scientist Presents Evidence for Belief, in which he describes his own conversion from atheism to Christianity. He also presents the case for an intellectually satisfying harmony between the worldviews of science and faith. As Vicki mentioned earlier, he is also founder of the BioLogos Foundation. So please join me in welcoming our esteemed guest, Dr. Francis Collins.
Well, thank you for such a kind introduction. I am not the book of Genesis, but I'm really glad to be with you this evening. And I'm delighted to be here at the annual ASA meeting. As you heard, I am a member and a fellow of this organization. It has meant a lot to me over the years. I must have missed the 40 years by 1 year, because I think I might have joined in 1979. So wait till next year, then maybe I'll get to stand up too. But, oh, I guess I got to stand up anyway. Thank you for that. There are so many people in this gathering that have been wonderful mentors to me over the ages that I've had the chance to get to know some of you. And I won't start naming them because I will leave somebody out and that will be embarrassing. But as people were being introduced and as I've encountered people coming here, it's just wonderful, again, to be in the mix of such deeply thoughtful, dedicated scientists who are Christians. And ASA has always been the kind of home that we could feel comfortable with. And it's wonderful to see this gathering happening again. The only thing I would say is that when we have the chance for the next gathering of people to be recognized, I'd like to see some more women. So let's work on that, ASA. Okay, that was an easy one, but I had to say it. So I want to talk to you this evening in sort of a two-part presentation. I am going to say a little bit about, from my own perspective, how these worldviews of science and faith can be joyfully complementary. And I mean joyfully, not just sort of rammed together in a way that you sort of had to make it work and you weren't quite sure it did. This is an opportunity for joyfulness. And I'll walk through a little bit of my own experience in that regard. But I also think, coming to you now as somebody who gets to stand at the helm of our nation's biomedical research, that it would be appropriate to talk about some of the issues, and one in particular, that we as believers need to be engaged with, where science and our view of what it means to be human are coming into an interesting space. So the second part of this will be a bit of a discourse about that. How should believers view the advances in biotechnology, and particularly something called gene editing? And I know you heard a little bit about that last night from Douglas Laufenberger, and I will get a little bit deeper into the thing called CRISPR-Cas, and maybe together we can think about what is the appropriate way to take this particular scientific opportunity, which I would think of as another one of God's gifts that science has made possible, and make sure we're using it in a fashion that would honor what it means to be fearfully and wonderfully made. So I will give a little bit of a biography, if you'll permit me, because people sometimes want to know, where is this guy coming from? Well, that's where I'm coming from. I was born on a small farm in the Shenandoah Valley of Virginia. That picture you see on the left is me at about age 8 or 9, standing at our farmhouse. And if you asked me at that point, what is it that you're dreaming of becoming, I would have told you, I have a career plan. It is to drive an 18-wheeler. I'm still waiting for that, by the way. And then I got interested in music and thought that might be a good way to enlarge my social circle, especially with people of the opposite sex. It didn't work that well, but I did learn a few licks on the guitar. And then I got interested in science because I had a high school teacher who taught chemistry and who got me inspired about the idea that science could unravel mysteries and that a scientist was like a detective, and you could approach a problem for which nobody had the answer and you could figure it out. And that seemed like something I wanted to do. So I went off to the University of Virginia and I got my undergraduate degree in chemistry. I ignored life science totally because it seemed very messy. It is, by the way, messy. I was right about that. I like the much cleaner world of chemistry, physics, mathematics. And having sort of learned to love that even more as an undergraduate, I went off to get my PhD in physical chemistry at Yale. My PhD was entirely done with computer cards and pencil and paper because it was a theoretical project. The title of my PhD thesis was Semi-Classical Theory of Vibrationally Inelastic Scattering. And when I take it off the shelf today, I can't believe I wrote that stuff, but there it is. But in the process of studying chemistry at Yale, I discovered there was some pretty interesting chemistry in life that I'd kind of ignored. And this molecule came to my attention, which somehow I had missed out on, that being DNA. And I realized, wow, this is not what I thought at all. I thought biology was just this infinitely descriptive science with no principles behind it. And what do you know? It does have principles, and it's digital, and it's satisfying. And you can even think about doing mathematics. And I realized I had a new calling, and that was to change my perspective to do something about life science, but I didn't know how to do that. So what to do? I wasn't sure. So I went to medical school. That's what you do when you don't know what else to do. It still amazes me those good folks on the admissions committee at the University of North Carolina accepted that as a reason to come to their school, but they did. And I immediately fell in love with medicine. It did feel like, okay, this is where I'm supposed to be. Yes, it's science, and yes, it's rigorous science, and yes, everything I've learned about chemistry and physics is going to help me here. But it also draws me into an area of biology and human medicine that seems pretty compelling. It was during that time, as a medical student, where I started out as an atheist, that I ended up becoming a believer at age 27. I'll say a little bit more about that in a minute or two. I loved medicine. I loved the opportunity to learn how to take care of patients, but I knew that I was really interested in DNA and genetics. So off I went after that to go learn the science of molecular biology and did a fellowship then in human genetics back at Yale in a different kind of laboratory. Having finished that, at which point you must think that I was at least 55 years old, but I wasn't quite. I finally got a real job and went to the University of Michigan to be there on the faculty to do what you do, teach medical students, run a research lab, take care of patients. And I loved every bit of that too. And my research at that point was to try to take the very rudimentary tools of human genetics and track down what were the actual causes of genetic diseases that remained for the most part unrevealed. Such things as cystic fibrosis or Huntington's disease. Diseases that we knew were inherited in dominant or recessive or X-linked fashion, but for which we had not succeeded in identifying the actual causative gene. This was tough. Young people in the audience must think, you must have been pretty stupid to think that that was a hard problem, because right now it wouldn't be. But in the 1980s, where there was no Genome Project, heck, there wasn't really much of an internet, and you had to basically sift around through 3 billion base pairs of the human genome to find one letter out of place. This was hard stuff. It was so hard that I felt I needed a visual image to say how hard it was, which is how I found myself in a Michigan haymow holding up a needle saying, "This is how hard it is." So don't give me a hard time that we haven't succeeded yet. And yes, that is a real chicken there in the haymow with me. I'm not sure where that came from. But ultimately, thanks to the opportunity to collaborate with other groups and bring amazing graduate students and postdoctoral fellows into this enterprise, we did manage finally to start to succeed. And maybe the first one of those that really convinced me and many other people this could actually work was cystic fibrosis. It was 1989 when we found, on a particular rainy night in May, a 3-letter deletion, a CTT that should have been there and wasn't, in patients with cystic fibrosis, in a gene that nobody had ever studied before, in the middle of a part of chromosome 7 that nobody had ever studied before, but there it was. And that was a pretty exciting moment to say it is possible to find that needle in the haystack, and this is something that ultimately might help somebody, because here's a disease that desperately needs new ideas, and which, by the way, I will say, in just the last couple of years, cystic fibrosis has moved forward to be a disease where we now have treatments that almost make this the kind of disease that should have a normal life expectancy. And it's incredibly gratifying to see that, even though it's frustrating to say it took this long, almost 30 years to get to that point, but we are pretty much there. Neurofibromatosis, Huntington's disease, other diseases I've worked on. More recently, my lab has worked on the most dramatic form of premature aging called Hutchinson-Gilford progeria syndrome. Children with this are born normal, but they age at about 7 times the normal rate. And even as their intellectual development is happening in a completely normal way, their bodies are failing them, and they die without treatment at about age 12 or 13. Again, it has been enormously gratifying to have a chance to work with these kids and their parents, and happy to say, as a result of that, found the cause of this, and now, just in the course of a few years, have come up with a treatment that prolongs their survival by at least 5 years, maybe more. So all of those are gratifying things, and it was a wonderful opportunity to have a chance to do so. And then I got this request in the form of a phone call from the person who was at that time the head of the National Institutes of Health, Bernadine Healy, saying, we want you to come to NIH and we want you to lead the Human Genome Project. The one thing my mother told me never to do is to become a government employee. So this was a little bit of a tough invitation to figure out what to do with, but my goodness, there was only going to be one Human Genome Project. In 1993, when I went there to try to lead the effort, it was only 2 years along. It looked as if there was a fairly high likelihood of failure, so there was a pretty significant risk involved. But if it succeeded, it was going to transform the way that we understand everything about human biology. And for me, having struggled for some of those difficult diseases to try to find the answers, this was the only way we were going to really scale that up. So I did get engaged in a way that led to a remarkable wild ride over the next few years, involving, I'm happy to say, some of the best and brightest scientists of the generation who decided that they were so excited about the historical potential that they too would take a chance and transform whatever they were planning to do and join this effort. Ultimately, it took 2,400 scientists in 6 countries, 20 laboratories, working 24/7 to make this dream come true. And in 2003, we did in fact deliver that first complete human genome sequence 2 years ahead of time and $400 million under budget. Yeah. So we had a nice party, and then we said, wait a minute, we don't know how to read this thing. It's written in this funny language that has just 4 letters in its alphabet, and we kind of can recognize the parts of the DNA that code for protein, but oh my gosh, that's a little less than 2%. What the heck is the rest of it doing? We've got a lot of work to do. So if we were going to take these discoveries and turn them into some better outcome than the one you see here, we had a lot of work yet to do about how to interpret the human genome, the instruction book for biology. And as a physician wanting to see the era of genomic medicine come true, this seemed like a pretty important next set of steps. So at NIH, we had the opportunity then, having figured out how to build these teams and how to give them bold goals to go after and watching them to see what they could do, inventing new technologies, delivering data sets that were put immediately into the public domain, which was the signature of the Genome Project, that everything was immediately accessible. Things started to happen. We developed a whole lot of other follow-on projects. This is just a few of them. And the genome began to make sense. Although I would say, to be honest, it still doesn't entirely make sense. We've got a lot of work yet to do to figure out how this incredibly elegant system, just 3 billion letters, can basically specify the biological properties of a human being, taking you from a single cell to what you are now. I mean, it is breathtaking to imagine the elegance of a system that can do that. You can't look at that and not be in awe. And more recently, we've begun to figure out how to take those advances even closer to human health. And if you'll permit me, I'm going to do a little advertisement here because it looks like this is an audience that could enhance this next project quite nicely. And that is this project which launched on May the 6th of this year, called All of Us. We want to figure out, how could you take everything that you could know about someone, sure, their genome sequence, but not just that, their environmental exposures, their health behaviors, their socioeconomic status, all of the things that happen in terms of their life choices, and put all of that together in a carefully secured database, but one that gives researchers access to the de-identified information. And let's not just enroll a few hundred people or a few thousand people. Let's enroll 1 million people. As of today, 88,000 people have signed up. We're on our way to 1 million over the course of the next 2 years. If you're interested in being part of this adventure, notice the URL at the bottom there, joinallofus.org. You might want to have a look. This is going to be a really amazing and transformative opportunity for us to figure out not just how to treat people who are sick, but also how do people stay healthy? Because we think a lot of the people that enroll in this are going to be just fine, and we want to figure out how do they stay fine. So they will get blood samples. There will be a lot of questionnaires. People will be invited to walk around with various wearable sensors, like one that I have on. I'm just curious, how many of you are wearing a Fitbit or some other kind of wearable sensor right now? It's getting to be a pretty good group of folks. Well, if you're in this study, you'll get one for free. So think about that. So that's the kind of thing that's now possible. And I'll say a bit more in a little while about some of the other advances that are happening in medical research. I was asked 9 years ago to come and be the director of the National Institutes of Health. I didn't expect that. I thought I was kind of done with the genome part of it and might do something else. But it's an enormous privilege to come back and stand at the helm of this enterprise. I did not expect to stay in that role. When there was a new election and a new president, there's never been an NIH director that was asked to stay over, but that seemed to have happened, and I'm incredibly fortunate and privileged to be able to continue in that role even now tonight. And I should have said at the beginning, that's a role that I play when I'm not speaking to the ASA. Tonight, I'm here as a private citizen. Did you hear that, government ethics people? Okay. So those are some of the things that have enriched my experience over the course of these years as a scientist. But now I want to talk to you about who I am as a believer. I'm very fond of this pair of images, which is evocative in a way that maybe words can't quite accomplish. This you can see quite readily on the left. One of those wonderful, amazing stained glass windows. I was at Notre Dame in Paris just a month ago, and there's one there, or actually 4 there, that are just unbelievably exquisite. But on the right is a biology image. Yeah, that's DNA. By the way, I have DNA in my tie also. I hope everybody noticed that. But the DNA you see there in the picture is looking down the long axis, which is not the way it's usually portrayed. But of course, it's a double helix, so imagine yourself looking down the long axis of a spiral. That's what it would look like. Beautiful indeed, and perhaps makes you think a little bit about beauty and the beauty of biology, the beauty of the world around us, which we acknowledge and admire and are moved by, and yet for which there's not an easy explanation about why it should matter. For me as a biologist, as a physician, the beauty side of science is something that always catches my breath. And I know that's true for many other areas. My goodness, those of you who look at those deep field pictures from the Hubble, that is an exercise of beauty, the most remarkable sort. So I mentioned that when I was a medical student, I ended up surprising myself by converting from atheism to Christianity. How did that happen? Well, first of all, why was I an atheist in the first place? I did not grow up in a home where faith was considered relevant. My parents were not necessarily insulting to people of faith, but they thought they were wasting their time. My father was a drama professor. My mother was a playwright. Our lives as I was growing up were about learning and education and music and theater, and faith was just not on the table. And so getting to college as an undergraduate and being in those conversations in the dorm where the local atheist was trying to upset the apple cart for the believers, I decided I kind of sided with the atheist in those arguments. The believers didn't seem to have very convincing responses. And I was also at that point, as many are, where it was convenient for there not to be a God that one had to be responsible to. It was much better to be able to call my own shots and not worry too much about that. And then when I went off to Yale to study quantum mechanics, it seemed increasingly to me that you could explain everything that mattered with mathematics. It didn't occur to me why that should work, but it did seem like this was sort of the reductionist model in the biggest way, and that everything that really mattered was down to second-order differential equations, which are, by the way, wonderful and beautiful in their own right. And so I gradually slipped from being what you might have called an agnostic to being an atheist. Not a hard atheist, but a soft atheist. And certainly if you had tried to have lunch with me and shared your faith, I would have been quick to point out to you that you were living in the past and that science made your particular perspective no longer sensible and probably anachronistic. But then I did go to medical school. And then in that circumstance, where this was not a hypothetical about issues of life and death, it was happening every day. And it was harder when I looked at people who were facing death to imagine myself and to imagine whether I'd really thought through what it meant to have a serious discussion about, is there a God? And if so, does that God care about you and me? And I had a particular experience with an elderly patient who had very advanced cardiovascular disease, who was a believer and whose faith gave her great confidence as she approached the end of her life, that God was there with her and that Jesus was taking care of her, and she would tell me those things and I would not really understand what she was talking about. But one day, after that kind of sharing of her faith, after she'd gone through a particularly dreadful experience of chest pain, she just turned to me and said, "Doctor, I've been talking to you about my faith, but you never say anything. What do you believe?" What an amazing question to be asked, and what an amazing setting. And in that moment, which is still burned into my brain, was the moment of realizing I have no idea how to answer that. And I should have a better answer than what I have. And I ran from the room feeling very uncomfortable and embarrassed and even sort of angry. How dare she ask me such a question? That's so personal. Well, yeah, she was being personal. She had the right to ask me too. And that began then this effort to try to understand, what do believers believe and why do they believe it? And is it, as I have assumed at that point, a fact that if you want to believe in God, you basically have to abandon rationality. That rationality will drive you the other way. That led me to the exploration of what people had written about this and what some of the faculty at the University of North Carolina Medical School who were believers would tell me about this. And it began to sort of make sense, but it really made sense when a pastor down the street pulled down from his bookshelf a little book and said, "You really ought to have a look at this." And that, of course, you've already guessed, was C.S. Lewis, and that was Mere Christianity. And reading through those pages about the path traveled by someone who seemed to have gone down the same road that I had as a non-believer who, kicking and screaming, the most reluctant convert in all of England, as he described himself, came to the point of realizing that it was inescapable to accept the truth of God's reality and of the reality of Jesus Christ. Well, that was not the answer I expected. And again, I fought it for a while. And I had my own opportunity then to debate with myself and with anybody else about why this was still an irrational decision. But ultimately, I realized that in my own experience as a scientist, there were a lot of interesting pointers here that I had ignored. Let's start with the big one. There's something instead of nothing. That's a hard question for an atheist to explain. You just have to say, well, it's not a scientific question, so let's move on. It is an important question. And science will, to my mind anyway, not provide an answer to that that's going to be satisfying. Those second-order differential equations, that describe the behavior of matter and energy. Maxwell's equations describing electromagnetism. Simply the inverse square law for gravitation. E equals mc squared. Why is it that nature should be defined in such an elegant way by mathematics? This is Wigner's phrase, by the way, the unreasonable effectiveness of mathematics. It is unreasonable. My gosh, it makes you think there's a mind behind all of this. And then, of course, there is the Big Bang, the fact that the universe had a beginning. Now, I don't want to get to the point of talking about all those other universes that are out there that may be big banging right now, because we could have an interesting discussion about that. But what we do know about our own universe is pretty compelling, that there was a start. And that seems to ask, well, okay, what made that possible, or who made that possible? And if you're going to say that required a creation event, then the creator could not themselves be limited in time and space, or you've just created an infinite regress and you've not solved the problem. So already here in these 3 steps, you've come to the idea of a creator who is not limited by space and time and who is one heck of a mathematician. And must also be one heck of a physicist. And this is the anthropic principle. Walter Bradley, I think you were the first person when I was struggling with what to believe in terms of science and faith, pointing out the anthropic principle and how it is that it is so incredibly unlikely that our universe could have the form that it does that allows complexity, because of the need for all of these constants that you cannot derive the value for by theory. They just are what they are. And if they were off by tiny parts, 1 in 10 to the 12th for many of those, you would no longer have a universe that had anything interesting about it. I'm not just saying you wouldn't have a universe that had carbon-based life. You just wouldn't have anything interesting. There would be no opportunity for complexity. The Big Bang would either go on into infinite space without allowing coalescence of anything larger than a particle, or you'd end up with a big crunch long before there was time for something interesting to happen, and on and on. If that's not an argument that you've tried out on non-believers, do so, because when I talk to Richard Dawkins, which I've enjoyed doing over the years, and I'm sure he enjoys poking at me just as much as I poke at him. But one time I asked him, Richard, what is the argument from science that you find most difficult to refute? And he said, oh, that fine-tuning thing, that one really bugs me. And again, your retort you will get from this, of course, is the multiverse. And it's a good retort on an intellectual basis. But where's the experimental evidence, folks? I'm still waiting for that. The alternative that seems to me a lot more appealing and requires less faith is that these dials were set. And that this amazing mind that created, back at the beginning, a universe that would follow mathematical rules also set those constants so that something interesting would happen. And then you have to say, well, is that a deism argument, or is that a theism argument? Is there something about what we observe today that says God is interested in us and not just interested in a nice, cool creation that happened a long time ago? And here again, much influenced by Lewis, the moral law, the experience that we all have of knowing what the difference is between right and wrong, an experience which has been true of all human societies down through history. Look at the appendix of Lewis's book, The Abolition of Man, if you're not convinced of that. People disagree, of course, about what belongs in the good column and what belongs in the evil column, and that's cultural. But nobody disagrees that there is a difference between good and evil, and we're supposed to do the good stuff. And we also know we don't do it. Well, goodness, if you were looking inside your own experience for some kind of a signpost that might point you towards a God who is not only a deist God, but a theist God, it'd be pretty interesting that you would find it in this very spot, pointing to a God who apparently cares about what is good and holy and wants us to as well. That, of course, then, as I went down those steps, began to make me realize how far short I was falling in terms of what that expectation would be. So, ultimately, over the course of those 2 years of struggling, I not only became convinced that atheism was the least rational of all the choices, as Chesterton called it, the most daring of all dogmas, because it's the assertion of a universal negative. But also that agnosticism for me was sort of a cop-out. It basically represented a failure to address the question. And then into that deepening uncertainty and unease came the figure of Jesus Christ. And for me, it was particularly critical at a time where I was beginning to appreciate that if there was a God who cared about us, it was a holy God, and I wasn't holy. And how was I possibly going to be able to have a relationship with the Creator that I was just beginning to glimpse? With that huge gulf between us, wouldn't that God be more likely to want to punish me than to love me? And then I realized what I had missed in much of this discussion was that's the whole point of Christianity, that Jesus came into that spot knowing all of us have fallen short ever since the fall, and taking upon himself the burden of responsibility, and on the cross, basically as a perfect sacrifice, making it possible for us to be washed clean, for us then to be able to approach God Almighty without the fear of being judged and with the certainty of being loved. And that's what grace was all about. And it was that realization that at age 27 led me on a wonderful hike in the Northwest in the Cascade Mountains to realize that my resistance was gone. And I fell on my knees on a wonderful dewy grass in the morning and said, that's it. I am at the point now of saying, I believe. And I want Jesus to be my leader. I want to be in that particular group of followers, and that will be the rock I want to stand on. And that was 41 years ago. And people who I immediately told about that thought it was amusing, and also suggested that I was on a path towards a brain explosion, because how could you possibly, as somebody who wants to study of all things genetics, which is going to lead you to information about origins, also be somebody who wants to study the Bible and be a believer? I got to tell you, 41 years later, I have not encountered an obstacle of any sort to putting those ways of knowing together. They are two ways of knowing. Yeah, I know it's a bit of a trite cliche, but it's kind of true. Science answering how, faith answering why. And we are given the privilege as scientists who are Christians to read both of those books. This is Bacon's wonderful analogy that I love to use, the book of God's words and the book of God's works. And if we do believe that God is the author of both of those, then how could they be in conflict? If we see a conflict, then surely we are the ones who have misunderstood the science or the biblical interpretation, or oftentimes both. But somehow we should not feel that as Christians we have to defend God against science. Isn't God the author of all that? And that has been for me a truly satisfying experience. I think I'm blessed that I didn't grow up in a perspective where the church was saying you have to believe a certain perspective about origins or your faith is at risk. And so many Christians in the past and in the present have been caught up in that. And I didn't have that burden to carry. So I could put together what I read in Genesis 1 and 2 and what I know from studying the genome in a fashion that illuminates and enlightens. Sure, there are plenty of areas of potential discordance. Who was Adam and Eve? Who was Adam and who was Eve? Those are big ones right now that I think are fascinating and important to talk about. But are they in fact salvation questions, and are they unresolvable? Or do we basically have a few options to say that could explain it, and then we can kind of move along and try to figure out what's the evidence one way or the other, and not have this fear that so many young Christians have that the whole edifice is going to come down because we can't find a way to put them together. I am confident we do have a way to put them together, and God gave us the grace to do that. But again, people are not all in agreement about that. And sometimes it's really troubling, and sometimes it's funny. And this is one of the cartoons that is particularly funny, where the goldfish are arguing. The cheddar one saying, we came from cheese, and the evangelical one saying, we just appeared in the bag. And sometimes the debates we have in our churches or in society seem almost this silly. So, in an effort to try to put down some of my thoughts about this, after not really talking a lot about this in a public way, it seemed like maybe the time had come, especially after a series of lectures I gave at Harvard back in 2003 as part of the Nobel Lectures, where I could see the hunger that Harvard undergraduate had to actually get engaged in this kind of conversation. They were just not having a chance to talk about science and faith. It was off the table. It was like a no-no, and they wanted to hear more about it and talk about it. After the book came out, I was deluged by all kinds of thoughtful questions. My email inbox was exploding with people who wanted to go deeper, who wanted to challenge some of the points, as well they should. And by the way, this book is really due for a second edition, and if I just had time, I would try to do that, because there are things that I would say differently now than I did then. But I couldn't possibly keep up with this. And then out of that, basically, was born an opportunity to try to address some of those questions. But basically, what the language of God says is a simple synthesis of science and faith. And I think, while this would not represent everybody in this room, I think you'll probably see resonance with parts of it anyway. And again, I think ASA has been a wonderful place for those kind of debates and conversations to come, both in meetings and in the journal. And here's mine. Almighty God, who's not limited in space or time, created a universe 13.8 billion years ago, with its parameters precisely tuned to allow the development of complexity and beauty over long periods of time. That makes a lot of sense to me from what we know from science. God's original plan included the mechanism of evolution to create the marvelous diversity of living things on our planet. Most especially, that creative plan included human beings, as we can now see, written into the DNA sequence of our own species, with all of its relatedness to other organisms that cannot be readily explained except by assuming a common ancestor, and that cannot be put together in terms of origins from two single individuals and none other. We have to accept that science is teaching us something different about origins than the most literal interpretation of Genesis 1 and 2, which, frankly, Augustine in 400 AD would never have embraced anyway. After God's plan for evolution in the fullness of time had prepared a sufficiently advanced brain, humanity was gifted with free will. Now, gifted, you can interpret it as a supernatural act, or you can interpret this as an intended, built-in part of the evolutionary process that God had planned all along. We were then given free will, consciousness, and a moral sense. And at that point, granted special status made in God's image. We humans used our free will to disobey God. Somebody said that the one Christian dogma that can be verified every day is the fact that we are all afflicted with original sin, leading to our realization of being in violation of the moral law. Thus, we were estranged from God our Creator, the same thing that left me with this sinking feeling, who embodies all that is good, loving, and holy. For Christians, like me, Jesus Christ is the solution to that estrangement. What could we call this synthesis? People have certainly called it various things. Many people over the decades have called it theistic evolution. I never really liked that term. Theistic was relegated to be the adjective. Evolution was the noun. It made it sound as if the latter was more important. I think evolutionary creation is a much better terminology. But I guess I invented this other term, bios through logos, life through the word, or simply BioLogos. And that was, in fact, the term that then was applied to the founding of this new foundation, God speaking life into being. It was just about 10 years ago that in the follow-up to all of those questions coming to me, and I know that I couldn't handle the responses and I needed help both from people who knew theology better than I did and who had better scientific background in various areas, that the idea of the BioLogos Foundation came into being. And along the course of those 10 years, so many people have come alongside with this effort, including especially Deb Harzma, who's now the president. And you'll hear from Jeff Schloss on Monday, who's the senior scholar for BioLogos, and many other folks here who have been engaged in this in very important ways that have greatly enriched the ability of this enterprise to try to help people who have very serious and legitimate questions, and always doing so in a winsome way that avoids the kind of mudslinging which all too often has characterized these conversations. So again, that was a privilege to be able to found, but when I accepted the invitation to come and be the director of the National Institutes of Health, In a memorable meeting with a phalanx of White House lawyers, I was instructed that doing that, one may have no other duties for any organization, no matter how different it seems to be than what the government is asking you to do. And so, beginning in 2009, I have separated myself from any formal involvement with BioLogos, but managed to watch from the sidelines and cheer wildly as others have moved this forward in ways that I think have been incredibly gratifying. So if you've not been to their website and seen the kinds of things that are going on there, please do. Again, BioLogos is careful not to say there's one answer to this particular thorny question. It's like, okay, here's some possibilities. Let's talk about what's on favor of this one and on favor of that one, in the way that I think is the way that we draw people together instead of driving them apart. And I do want to give a particular shout out, because we're here at Gordon College, and we're here in this remarkable space that I've had the privilege of being in previously, and give a shout out to Judd and Jan Carlberg. Jan, who's sitting here in the front row. Judd, who is the president of Gordon College, was such a remarkable visionary in terms of how science ought to be a critical part of the college curriculum, and has been the case here, and has been even further enhanced, as I found out this afternoon by talking to President Lindsay, by increasing the number of science majors to now to 26% of their undergraduates. And Judd also, after stepping down as president of Gordon, stepped up as chairman of the board of BioLogos. And I learned so much from him. And I learned so much from Jan. And I continue to learn from Jan as she writes wonderful words in her blog every few days about life experiences and how they all fit together. We lost Judd a couple of years ago to cancer, but his imprint on what happens in BioLogos, and certainly his imprint on me, will go on. His legacy here at Gordon and in many other ways is a lasting one. And I had to give a comment about him, especially standing here in this space. So what about this issue of science and faith, and how do we bring it together? Obviously, this is a place where many people have encountered challenges, and I am very fond of Augustine's words here about, in essential things, unity; in non-essential things, liberty; in all things, charity. BioLogos is really trying to stand up for that, especially the charity part. So that was the first part of the talk, and I hope you'll forgive me for having been pretty personal there, and maybe even a bit autobiographical about the path that I've been on. And everybody in this place has been on interesting paths, and I've learned many of them by talking to you. And we should be sharing those things with each other, to encourage each other, to learn from each other, to recognize that we are a community that has a lot to share and a lot to give. But now I want to challenge you a little bit about where we're going in a particular area of science that I believe is one where Christians need to be in the room and at the table as we begin to debate potential boundaries of what science makes possible, but what is possible isn't always wise. And this is an area which I am both enormously excited about because of its potential to alleviate suffering and save human lives, but also where there are some concerns about some of the possible applications. There are, of course, many ethical challenges in biotechnology I could talk about. Privacy of information. I invited all of you to join all of us and give us all of your electronic medical information and your DNA sequence, and you better be asking questions, I would think, about how is that going to be protected. We could talk about animal research and what are God's call to us in terms of our responsibility as stewards, especially when it comes to non-human primates. We at NIH have decided not to do research on chimpanzees of an invasive sort anymore. Neuroethics. The Brain Initiative is going to figure out how those 86 billion neurons between your ears do what they do. And in just the next 10 years, we're going to have insights into the functions of the brain that are going to be profound. Maybe even ways to figure out how to transfer memories from one person to the other. Maybe in the long run, ideas about how to download your consciousness in silico into a machine. We need to think really hard about what that means from a perspective of a Christian believer. Human embryonic stem cell research, obviously a hot topic, as it should be, and it has not gone away, although much of the debate has now, I think, dimmed a bit because we can make stem cells out of your skin or your blood, and we don't need to depend on embryos to derive them in the way that we did for a while. Human-animal chimeras. Some of you are going, oh my God, you're not doing that. Well, think about it. If you want to create for somebody who needs a heart transplant, their own heart? Well, one way to do that, take a skin biopsy, take those skin cells, turn them into pluripotent stem cells, the induced pluripotent stem cell, they're still your cells. Inject your cells into a pig embryo. But the pig embryo has been designed so that it has a genetic defect so that that pig cannot make a heart. But your cells could. Okay, if there's a pig that's live born, it's got a human heart. Otherwise, it wouldn't make it to that point. There you go. There's your transplant. Is that okay? People are proposing those experiments. We at NIH will not at the present time support them because I think there are serious ethical questions about, wait a minute, where else did your cells go? Are they in the brain? What is that pig thinking right now? Got a little work to do on that one. Dramatic life extension. All of us would like to see our health span extended, where we have the chance to live out a full life without being afflicted by chronic disease. But what about extending survival from the current max of 120 years to, say, 500? Is that possible? I don't know. It might be. Is that a good thing if it were? We've got to talk about that too. I can talk about any of those, but I'm going to talk about the last one, which is gene editing, and see what you think. And that means I need to get into this technology called CRISPR-Cas9. This came out of the most interesting and unexpected place, and it's a wonderful way to tell you, if you want to support science, don't just support science that's targeted to a particular disease, because you never know where the big exciting things are going to come from. This one came from yogurt, and the study of viruses that attack bacteria seemed like the most irrelevant possible kind of science. Why would NIH support that? Well, they discovered this whole system which bacteria use to fight off viruses that attack them. And bacteria basically have a memory of what the nucleotide sequence was of viruses they've seen before, and they know how to go and cut that, which means they have to have an enzyme that does the cutting, and they have to have some kind of a template that tells them what to look for. And that's what CRISPR-Cas9 is. Cas9 is the enzyme. In order to work, it needs what's called a guide RNA to go and zero in on a particular sequence of DNA and say, cut here. It's revolutionized basic molecular biology because it works in any cells, including human cells. And it's easy to do. Any laboratory doing molecular biology has started doing CRISPR-Cas9, and it works the first time, which nothing ever does in molecular biology, but this does. And while this is, after all, recombinant DNA, when we've been doing recombinant DNA now for 45 years, this is just incredibly efficient and incredibly specific, so that you could, and there's still arguments about whether we're there yet, apply this to a human cell where you wanted to go and change one letter out of 3 billion and do nothing else, and you could do it. That kind of precision was way out of reach until this came along. And now we have the chance to apply it to human genetic disease, which seems pretty darned exciting. So, this is my blog, by the way. If you're not reading my blog on Tuesdays and Thursdays, you can do so. Simply go to the NIH Directors page, and every Tuesday and Thursday I try to write about something that's happening scientifically. And we at NIH are pushing pretty hard now to try to speed up the process of applying gene editing for benevolent purposes to treat various diseases that desperately need answers. So there's a big promise here. Welcome to the golden age. New England Journal, gene editing of a gene called CCR5 to treat HIV. Looks pretty exciting. Even delivering it to somebody with a genetic disease, this individual with Hurler syndrome, where this is injected in vivo to try to do something about it. We don't know whether it's working or not. And also an application that FDA has approved to bring this therapy for cancer, where you basically edit the immune cells to teach them what to go after. The promise, but the peril. Schweitzer, we must not allow our technology to exceed our humanity. Let's think about that. And now you see that these headlines are not about treating immune cells. We're talking about treating embryos. And science calls this maybe a weapon of mass destruction. Okay, clearly something to talk about here. Now first, let's admit that playing God is not a new thing for biotechnology. It's been around. Recombinant DNA, when it came out in the 70s, was subjected to many of these same discussions, and we should go back and learn from those and not have them all fresh. Genetic engineering, kind of the same as recombinant DNA with a different name. Cloning, remember that? The Human Genome Project was considered to be potentially playing God, reading the instruction book. Synthetic biology, where you don't just read a genome, you write one. Genome editing, which we're talking about now, maybe even do-it-yourselfer genome editing in your garage. And again, are we ready to play God? And people tend to go to the Sistine Chapel and choose images from there to illustrate their point. So this is not a new concept, but where are we with this one? Why is this different? I think it's different because it is so incredibly efficient and so easy to do. Much of our conversations in the past have been theoretical. It's not theoretical anymore. The Vatican's interested in this. I was there in April for a 3-day conference about this very topic. The Pope addressed us in a very thoughtful way about this whole circumstance. I gave a lecture surrounded on both sides by cardinals, so I haven't done that before. And actually, it was really gratifying to see the Catholic Church very interested in learning more about this and concerned that the Catholic Church has often been portrayed as just saying, no, don't do it, and this time trying to understand the pros and the cons of various types of applications. So let's separate it out here, because this is where things get blurry if you're not careful. And that means I need to take a minute to describe the difference between somatic cell gene editing and germline gene editing. A somatic cell is a cell in your body that's not going to be passed on to your children, which is most of the cells in your body. That means one could modify those without having a consequence for future generations. We could get your consent if you wanted it done, and if you decided you did, we're not necessarily crossing an unprecedented ethical line. As opposed to, if we're talking about modifying germ cells, sperm or eggs or embryos, then if something goes wrong, it's not just that person, it's their future offspring who are not there to give consent, and you've created a very different scenario. But let's talk about the somatic cell part, because here is where I think the incredibly positive excitement resides. This would be focused on a particular organ. It has the promise to cure a lot of genetic diseases. It's already being used, as I showed you in a couple of those headlines, for cancer immunotherapy. Basically take out the immune cells, educate them with some gene editing about what they should be doing, give them back and have them go after that cancer. This is leading to dramatic results. People with widely metastatic cancers who we thought had weeks to live, who after this kind of gene editing cancer immunotherapy are back at work. And they don't just appear to be responding, they're cured. This is unheard of, and all of this becoming possible because of the ability to activate the immune system to go after those cancer cells that somehow they had not noticed, and now they notice in a big way. And then sickle cell disease. This is one of my dreams. When I was a postdoc, it was diseases of hemoglobin that I worked on. And here's sickle cell disease, which was the first molecular disease described in 1910. Inheritance shown to be recessive in 1949. The genetic basis determined that this is a misspelled amino acid in the 6th codon of the beta globin gene. 1957, the gene's cloned in 1980. There is a drug, hydroxyurea. It's helpful, but it's certainly not a cure, and it has a lot of side effects. Bone marrow transplants have now been applied, but unless you have a match, which only about 15% of patients do, you're not a candidate. And this is a horrible disease for those who are afflicted. The bone pain crises that you have, sometimes many times a month, are about the most excruciating kind of pain that humans can experience. And virtually everybody with sickle cell disease ends up in trouble with opioids, because that's the only way to get any kind of relief. And then they're treated, oftentimes by the medical care system, as the kinds of people you don't want in your ER because, you know, they're addicted to opioids. Well, you would be too if you had this condition. We need to come up with an answer, and here is our chance. Gene transfer with viral vectors has been tried, but CRISPR-Cas gene editing, to my mind, is the answer. And we will see, by the end of this calendar year, the first clinical trials. Now, how do we do that? Sickle cell disease, it is a disease of red cells. Where are red cells made? In the bone marrow. There are stem cells in the bone marrow that make all of the blood cells. So take out the bone marrow, sort it to find the cells that are those stem cells, use the magic of CRISPR-Cas to fix the sickle mutation, that one letter that's wrong. And there's an easier way to do that I won't go into, which would be even more readily applicable. Expand those cells that have been corrected in the laboratory, and then make a little space in the bone marrow, because you have to have a place for the new cells to go, and there are ways to do that with sort of a gentle version of chemotherapy. Give them back. This has been successfully done in mice. It's been successfully done in non-human primates. I think it will be successfully done in humans before the end of this year. And I would think 5 years from now, anybody with this disease ought to have a chance to be cured. Now that's a dream, and a dream that would only be possible because of these advances. So you got to say, this is a good thing. And that's one of those molecular disorders, but how about all the others? There are approaching 7,000 disorders where we know the specific genetic misspelling. How many of them have a treatment? 500. If we're going to find treatments for the rest, we probably don't want to do it one at a time. You want to come up with a general, scalable strategy. This might be it. If you could figure out how to deliver Cas9 and an appropriate guide RNA in vivo to the organ where the disease is happening and fix the mutation, then you'd have something remarkably scalable. That's one of our dreams. That's what NIH is now putting an extra $160 million into, because that's harder. With sickle cell disease, we're talking about taking the cells out where you can sort of keep a track on them, do the correction, make sure you did it right, give them back. If you're trying to treat somebody where the disease is in the brain, that's a lot harder. But it's not impossible. So this is a reason to celebrate the advance of gene editing. But what about the other part, the germline part? So it does involve, in that case, changing the DNA of a human embryo. That means that affects future offspring. Frankly, I have a hard time identifying medical applications where you would want to do this, where you don't have other alternatives to basically avoid having a child with a terrible genetic disease. One can do that in other ways with pre-implantation genetic diagnosis. And you'd sort of have to do that anyway if you were proposing to do gene editing on an embryo. And of course, it opens the door to going beyond something you would call medical to something you might call an enhancement. Are we ready to go there? This is the nexus right now of the intense debate. So once those concerns and academic discussions are more real, which they now are, what do we do? Again, the area of greatest concern, improve gene editing tools for germline modification. Again, I don't think the specific examples for prevention of disease are particularly compelling. And what about enhancement? First of all, I've told you that CRISPR is incredibly precise, but do we really know that down to the full detail you'd want to? So you thought you were fixing this gene over here. Is there any chance you actually tweaked that one over there unintentionally? We haven't solved that yet. But maybe more importantly for this audience, what about the philosophical, theological concerns? Is this treading into territory that goes beyond what we as humankind should be doing and into God's territory? Now first, let me argue, in case people are saying, well, it ought to be okay for medical purposes, but not for enhancement. I'm going to say the boundary between treatment and enhancement is really hard to identify. And so it doesn't help you that much to try to draw that bright line. It's not bright. Let me give you some examples. Look at this diagram here. On the y-axis, we have time between what we can do now, what we could do in 10 years, what we can do in 100 years, and maybe what's impossible. And on the x-axis, we have my own personal judgment about whether a particular application is going to be admirable, acceptable, questionable, or just unacceptable. And what would we want to put there? Well, I'll throw a few things down. Hair coloring, just to start off. Okay, I'm going to say that's acceptable. I'm not sure it's admirable, but it's acceptable. And it's certainly available now. Music lessons, okay, that's an enhancement. If you're somebody who took music lessons intensively before age 7, I can see that in your brain. You've actually enlarged that part of your brain visibly by the kinds of scans we can do. So that's an enhancement of your neurological function. But we would call that admirable, at least I would. Immunizations. You are changing the DNA of your immune cells when you give a vaccine. That's a genetic modification. But we think that's a really good genetic modification to get those immune cells ready for the time they encounter a really bad pathogen. So admirable and available now. Okay, prayer. Maybe I'm stretching a little bit to call that an enhancement, but I hope it is, especially when I'm having a bad day. So prayer, admirable and available, for sure. Okay, Viagra, we could say, you can decide whether I've got that in the right location or not. Probably depends on how old you are. And Ritalin. If it's Ritalin for attention deficit disorder, for somebody who's really badly afflicted by it, okay, that has certainly helped some kids, although one can argue it's being much too widely applied. But Ritalin for college students taking exams, no, sorry, that does not seem like something that we should be supporting. Similarly, drugs to treat truly morbid obesity instead of having people go through bariatric surgery, that would probably be a good thing if they really worked. But drugs to keep normal people thin, that doesn't feel quite right. Now maybe you say I just made a distinction between enhancement and treatment, and you could decide where your boundary is. Is it when your BMI is 30? Or above, then it's a treatment, but if it's 29.9, it's an enhancement. You can get the sense here. This is a blurry thing. I talked about chimeras to produce transplantable organs. Well, you could say this could be an answer to our shortage of transplants, but we did have that what's the pig thinking moment, so we have to think about whether we're ready to go there. On the other hand, if you could build a heart in a dish with your own cells, and we're starting to get closer to that, using organoids that you can grow in various kinds of tissue engineering experiments. Maybe that would be not a bad thing. Gene editing to cure sickle cell disease, again, I'm saying we ought to be there in 5 years, and that's a really good thing. To cure thousands of other rare diseases, if we could figure out how to extrapolate that, I think that's a really good thing, as long as it's somatic. But human embryos to prevent disease, I don't think we are ready to take that on. And certainly to enhance performance, I'm going to put that way over there on the right, because that does seem to me to be getting into territory that we ought to think hard about. Who decides what's an improvement? Think about that. And what does that say about equity and justice and who has access to these kinds of things? Extension of lifespan to 120 years? Well, that would be an enhancement. We might get there in another 20 years. To 500 years, I don't know whether it's possible, but it would raise all kinds of questions about whether that's a good thing or not. It certainly would wreck Social Security. Download your consciousness to a computer. Well, I have a lot of questions about that. And then again, designer babies with precisely predictable outcomes. I'm going to say that's not only not acceptable, it's never going to happen. Because there's a fundamental flaw, and people put forward those scenarios, although they make good Hollywood movies, and that is the idea that you could dial in the nature of an individual by getting their DNA sequence exactly the way you want it. They would forget, if they tried to do that, that the environment is probably a lot more important in terms of what kind of person that turns out to be. So the yuppie couple that dials in the perfect genome and discovers that they have a sullen 15-year-old who's up in their room smoking pot because they forgot about parenting would discover that this is not a scenario that you want to support. So again, let's, as we have this debate about germline alterations, admit that enhancement versus treatment is a tough line. I'm going to say for me right now, neither one seems like a line that we should be crossing, and NIH at the moment will not support human germline editing. When we get to these kinds of questions, people always ask, well, will Christians and non-believers end up in different places? Sometimes yes, although a lot of times we do end up in the same place, because folks, the basic principles of bioethics are based upon Judeo-Christian principles that have been around a long time. Respect for persons, beneficence, that you're trying to do good and not harm, justice and equity. Where do those all come from? They come from the Bible, and they are shared by other world faiths as well. So we don't have to worry if it's coming down to those things, are we going to have profound differences But when it comes to such things as the dignity of the human being and the exceptional nature of humanity as being created in God's image, then maybe we do have a special circumstance. The implications of gene editing can be profound. The safety risks are unknown. Future generations will be affected but aren't consenting. Who decides what's an improvement? If it actually provides benefits, what happens to equity and justice? Will there be a further separation between the haves and the have-nots? And what does it mean to be human, if that can be fundamentally altered by tinkering with our biological instruction book? How does this affect our relationship with God the Creator, the Imago Dei? Are we going to be fearfully and wonderfully made all over again? So we have to look at Psalm 139. For you created my inmost being. I don't think that phrase is written to CRISPR-Cas. You knit me together in my mother's womb. I praise you because I am fearfully and wonderfully made. Your works are wonderful. I know that full well. My frame was not hidden from you when I was made in the secret place, when I was woven together in the depths of the earth. Your eyes saw my unformed body. All the days adorned for me were written in your book before one of them came to be. Those are powerful and poetic words, and I think they're very relevant to the circumstance that we now face in terms of where the line should be drawn. Now, the good news is the scientific community is paying attention to this. The National Academy of Sciences put out a report on human genome editing, science, ethics, and governance, basically saying we should not be doing human embryo editing for enhancement purposes. Again, they were a little unclear about what that boundary would be. They did kind of open the door a little bit to the idea that there might be a justification for doing it for medical purposes, and without really arguing why that would be necessary very clearly in particular cases. So there is some attention here to this. And again, NIH, doing what we can, are saying we're not ready to go there. But that isn't happening everywhere. Things that are not supported by NIH are certainly going forward. The Chinese in particular have been doing human embryo gene editing, not so far in a way that tries to reimplant the embryo and result in a human being, but they have certainly shown that this works in human embryos, and then they sacrifice the embryo after a few days. And it works. So there's no sort of, oh, well, God will prevent this somehow from being successful. It does look as if this is a possibility. All the more reason why we really need to think about it. Now, you might then say we should basically put a strong stop on a lot of this that's going on in terms of genetic alterations. Again, I hope the distinction between somatic cell and germline was clear. And I want to come back one more time to say that what's happening in genetic therapies for somatic cell purposes is something that we as Christians should celebrate and encourage with all the appropriate guidance about ethical principles and informed consent and risks and benefits. Because there's such cool things happening here. And I got to tell you about one more, and then I'm going to be winding up here. Spinal muscular atrophy is one of the most heartbreaking human conditions that you'll ever see. These children are born floppy because the cells in their spinal cord that innervate muscles, the motor neuron cells, are affected. They basically have ALS as infants. It's a genetic disorder. We know exactly what the genetic defect is. It's not that uncommon. And these kids can never hold their heads up, and they die by about age a year, basically from suffocation because they can't breathe. It is one of the most miserable and painful things that you can imagine to watch your child go through this. Well, we know what the defect is. Could we actually compensate for what's missing there? I did not expect that this was one of those that we would have an answer for any time in my lifetime. But a team of scientists, in this case at Nationwide Children's Hospital in Columbus, Ohio, working first with an animal model and then extending this with full approval to kids, basically developed a gene therapy strategy, which is given one time as an intravenous infusion with a particular vector that finds its way to the spinal cord in a remarkably effective way. And these kids, who were identified as early as possible, then received this. And we began to hear noises that some of these kids looked like they were actually holding their heads up. And then I heard some of them able to sit. And I was recently in Columbus, and most of this hasn't yet been published, but I met a couple of the kids, and I got a quick video of one of them And so here is a little boy who has this condition, who should by now have not survived because he's almost 3. He's not just, yeah, isn't that amazing? Yeah, he's going to work, got to push the elevator here. Standing on his tiptoes. I mean, for a kid who you'd not expect to be alive. And here he is with his dad on the monkey bars. Just unbelievable. If that doesn't give you chills, I don't know what will. So we're on a really wonderful roll here. And the genetic approach is starting to work. And it's taken a long time to get it there. And yet there are these important circulating ethical issues. And we have to be sure that we get that balance right. So how do we do that? Well, we have to be sure we're bringing hope to those who are suffering. That's an ethical imperative. To stop research that might have helped that little boy would not have been the answer. I don't think anybody would propose that. Doing nothing is the most unethical of the options. So we have to proceed, but proceed responsibly. Long-standing ethical principles shared by believers and non-believers can form the cornerstone, but international consensus would be desirable but difficult to achieve. There is no international body that can enforce such a consensus. People of faith, all of you who know the science, have an important role to play. We need to be more engaged than ever in these kinds of conversations about where boundaries ought to be set. And I'm privileged to be part of ASA, where I have those connections and I use them, and privileged that BioLogos is also in a space to be able to start those kinds of conversations so that they can be channeled in a way that helps come up with the right conclusions. Finally, this is portrait that hangs over my desk in my office at NIH. It was given to me by a artist who painted this in her mid-80s. She's a professor of art at James Madison University. And you might wonder, what is this picture? Well, her title of it is, This is Jacob's Ladder. And interesting that she painted this where the ladder is that double helix. With angels going up and down on it as Jacob is learning something about what is God calling him to. I think this is a nice metaphor for what we try to do as scientists who are believers, to bring together the awesome things that are being discovered in science and remember really what it's all about, which is trying to figure out how to live the way that Jesus called us to do. For me, Jesus spent a lot of time healing people. I think, therefore, we were called to do that too. But we have to do so in a fashion that represents and adequately stands up for all those principles that we were also given as human beings, but especially as human beings who have the image of God in our head and our mind and our body. So just one word of sort of caution here, which comes from Lewis, my favorite writer, about where we might end up if we're not thoughtful about this. And if you haven't recently reread The Abolition of Man, it comes along at a pretty timely moment. If one age really attains, by eugenics and scientific education, the power to make its descendants what it pleases, all men who live after it are the patients of that power. They are weaker, not stronger. For though we may have put wonderful machines in their hands, we have preordained how they are to use them. What a thoughtful and somewhat worrisome way to think about where we're going. So, again, I think we have the opportunity as scientists and believers to have significant influence, to both celebrate the advances and also sound alarms when those advances are getting into territory which is not what should be the right answer for God's plan for us. And I'm delighted to have a chance to be here with all of you in this meeting to talk about things like that and many other things as well. And with that, I will thank you and be glad to take a few questions. Thank you all. Well, Chekhov said that when you go to a play and there's a gun on the table in the first act, somebody's going to use it before the play is over. And this is not a solo. This is a wonderful rewrite of a familiar hymn tune that I think is probably the best hymn that's ever been written for an audience that cares about science and faith. And the words by Thomas Stroger, who's a pastor in Colorado, and praise the source of faith and learning. And let me ask the person who's running the PowerPoints, because it's hard for me to push the button, can you help advance when we come to each of the stanzas? And you could start right now. Why don't you stand up and sing with me? You'll know this tune. Praise the source of faith and learning that has sparked and stoked the mind with a passion for discerning how the world has been designed. Let the sense of wonder flowing from the wonders we survey keep our faith forever growing and renew our need to pray. God of wisdom, we acknowledge that our science and our art and the breadth of human knowledge only partial truth impart. Far beyond our calculation lies a depth we cannot sound, where your purpose for creation and the pulse of life are found. As to currents in a river, find each other's undertow, till converging they deliver one coherent steady flow. Bend, O God, our faith and learning till they carve a single course, till they join as one, returning